I think we're going.
Okay, perfect. So in order to determine the deletions or mutations on specific chromosomes, scientists use three main methods, and one of these is PCR. And PCR is a technique that is used to amplify small segments of DNA. And this process can be completed as many as 30 to 40 times. And it is important because significant amounts of DNA are needed for analyzing genes. And this would not be impossible without PCR amplification. And since tumor cells produce unique mRNA transcripts depending on the type of cancer an individual has, RT-PCR is used to determine which serves best as the biomarker for a particular cancer cell type. And this is used in labs to study gene expression as well as to diagnose genetic diseases and monitor drug therapy. And a third commonly used method is genetic sequencing. And this is used to determine the order of the four bases that make up the DNA molecule, which is able to tell scientists the kind of information that is carried out in a particular DNA segment. And this is done with the use of electrophoresis to separate the DNA fragments based on their size, and it allows them to be sequenced at a much lower cost and higher speed. And it also enables testing to be done on multiple different genes from multiple different patients at the same time. So research throughout the years has revealed many different genes that are prone to mutations. And there are four types of mutations that are associated with receiving a cancer diagnosis. The first one is copy number alterations. And these are changes to a chromosome structure that result in a gain or loss in copies of sections of DNA. And another one is methylation. And this is a process by which a methyl group is added to a DNA molecule that affects gene expression. And the third one is impaired expression at the mRNA or protein level. And finally, there are translocations, and these happen when there is a break in a chromosome, causing it to fuse with a different chromosome. And so all of these genomic alterations result in point mutations, which is when a single nucleotide base is either inserted or deleted from a DNA sequence. And just as a little fun fact that we thought we should add, solid tumors exhibit between 40 to 150 non-silent mutations, which are mutations that have an observable effect on an organism's phenotype. So there are many different benefits to knowing a specific mutation, and one of these benefits is that early detection leads to a better prognosis. And another benefit is precision treatment, which provides personalized medicine for the treatment of cancer based on the genetic alterations of an individual's disease, which provides basically an all-around better prognosis. So tumors may be different in their sensitivity or resistance to a certain gene form of treatment. And one reason for this is because many different factors within the environment in which the cancer cells exist contribute to how well a drug will work. And these factors vary between patient to patient, as well as tumors within a single patient. And also mutations on certain genes are more likely to provide an individual with a poor prognosis. And since cancer cells have multiple different mechanisms for surviving and growing, combining treatments that have different mechanisms of action may kill more cancer cells and reduce the chance that drug resistance will occur. And if an individual has a mutation, they are able to make early lifestyle choices. And these are changes, and these can be just as simple as like coming in more regularly for screenings. So these are some stats on the benefits of early screening. So there's been a 70% decline in cervical cancer cases since the Pap smear came into use. And the 5-year survival rate for colon cancer is 91% for those with early stages and only 11% for late stages. And for breast cancer, it's 99% for early stages and only 29% for late stages. And it's 100% for the early stages of prostate cancer, while it's only 31% for the late stages.
So this first piece that I want to talk about, it's not particularly new, but it's super relevant to cancer geneticists. It's the Cancer Genome Atlas, which is a project by the NIH that was started in 2006. This helps researchers who are looking for genetic biomarkers for cancer screening with just providing a large database of information. It's 33 different cancer types from thousands of patients worth of genome sequencing just regarding potentially what caused their cancer genetically. This has helped support the field of computational biology by allowing researchers to perform these dry labs to find data correlations and connections between certain gene alterations and cancer types. And the best part is it is publicly available to all researchers who are looking at cancer and genetics. So one important note is all the following techniques are used for diagnostic and they're not necessarily predictive. So this is not like the genetic testing for cancer that runs in a family, which could indicate the likelihood of developing a certain cancer, but instead screening for active cancer with various genetic measures. So the first measure is circulating DNA or ctDNA. This is DNA segments that are present in the bloodstream. They are tumor derived and they are released via apoptosis, necrosis, or secretion. These are analyzed via next generation sequencing, which we've talked about previously, or with allele specific PCR, which I'm going to talk about in just a moment. And it's thought that these are present in very early stages of cancer, so they could be really helpful to diagnose cancer before it could be visualized with other methods. So this is allele-specific PCR. It's basically the same as regular PCR. It's just kind of done twice so that you can use two different primers, one for the common allele and one for the specific allele or the mutation. So if you know that you are looking for a specific allele mutation, you can use that primer and find it through the process of PCR. So some other uses, some other benefits of ctDNA is that it's measured quantitatively, so it can suggest a prognosis at diagnosis. Based on how much is present. You can monitor for relapse, where imaging might not find tumors until it's too late. And you can track the course of disease, assess response to treatment. A lot of the current methods for doing this use radiation, like CT, which you see on the left. And so we know that the CT scan exposes you to radiation, and radiation exposure can lead to more genetic mutations, which is not ideal for cancer patients who may already have mutations. So the next measure is microRNA, which are also circulating in the bloodstream, but these are RNA and not DNA. These are also a little bit different from ctDNA because they're packaged into exosomes. And these are very stable because they're not degraded by enzymes or changes in pH, so they can live in the blood for quite a while. They can also withstand temperature changes, going from hot to cold, back and forth, which is really useful for lab and clinical settings where a sample might need to be put in the fridge, taken out, heated up, back and forth. Because these are RNA and not DNA, you have to use reverse transcription for PCR, but this is a pretty common practice, so that's not much of a problem. And these are also released via apoptosis, necrosis, or secretion. So the predictive power of these is pretty high, and it's suggested that some cancers could be predicted up to a year before an individual would receive a diagnosis with other screening methods. These are highly specific and highly sensitive to the testing methods. As you can see on the left, that black line just indicates that as specificity increases, even at low amounts, it's very selective for the specific RNAs that we're looking for. And if this works consistently, it's really promising for early non-invasive cancer screening. So another method is changes to promoters. Some promoter regions are highly methylated for certain genes. As you can see, not all of them, but those first three are showing really high levels of methylation in the promoter regions in the tumor and in the areas surrounding the tumor. And this just suggests that the role of regulating genes in tumor development could be important. And this is relevant because some cancer screening uses whole exome sequencing. And in this case, the exome does not include the promoters. And so that would not be very useful here. Finally, I just want to talk about microbial DNA biomarkers for cancer. This uses that cancer genome atlas that I mentioned earlier. In this study, they took all of the human DNA from those samples and threw it out and just kept the prokaryotic DNA and basically studied the correlations between microbial DNA markers and cancer. And so those maps on the right are heat maps showing a correlation, basically. So the darker the color or the more red the color, the hotter the color, the higher the correlation. So the first map shows one cancer type versus all other cancer types, and you can see there's a lot of red. So that means there's a lot of correlation between certain markers and specific cancers. And then B shows only early stage cancer. So again, you can see a lot of that dark red, which is really promising. And then C and D show detection of cancers without known genetic mutations already. So this is great for cancers where we don't quite know what's causing them. And just kind of interesting, the people who did this study actually founded a startup called Micronoma, which offers a liquid biopsy for cancer screening, basically a blood draw, and then they look at that and try to match it to these cancer biomarkers.